Hello guys, good afternoon. I am starting the immune system part 1 today, which is chapter 15. I will go over defense mechanism, which is two types. We call it innate or non-specific and adaptive or specific immunity. In the specific immunity, we're going to talk about function of B lymphocytes, which produces antibodies. And we also talk about the function of antibodies. Uh, Another cell responsible for the specific immunity is T lymphocytes. So we're going to talk about their types and functions. And at the end, we're going to talk about active and passive immunity. And what is the mechanism behind inducing active and passive immunity, okay? So the immune system and lymphatic system is highly dependent on each other. That's why we always talk about lymphatic and immune system together. Here you can see the diagram. This is the cardiovascular system, which is the heart and blood vessels. When heart pumps blood, it ultimately becomes capillaries. And then it is capillaries where we exchange gases and nutrition. So gases are going outside and nutrition is also exchanged here. But when these capillaries becomes veins, not all the fluid or exchange material return back to the veins. Some of them remains in the tissue. And that is called tissue fluid, which is in your body part everywhere pretty much. And those tissue fluid are excess fluid which is escaped from your capillaries. And that tissue fluid is carried back to the heart through the lymphatic system. So when lymphatic system get excess fluid, old cells, extra protein, or any microorganism in the tissue, that is also carried through the lymphatic capillaries. And then in the lymph nodes, where we have a lot of white blood cell, they are cleared, killed, modified, detoxified, and then it returns to your heart, the fluid, okay? That's how the lymphatic system is carrying lymph as well as also playing the defense mechanism of your body. That's why we always learn lymphatic system and immune system together, okay? Let's start with the summary of immune system. And then I will go today all about the non-specific or innate immunity. And in next lecture, I will talk about the specific immune system, okay? So in the right side here, we have innate and we have acquired. So basically immune system is all the structure, function, which help in defending us from microbiological damage or which protect us from infection. And this immune system are two types, the innate immune system and the acquired immune system. Innate immune system is permanent. We born with it. That's why innate immune system is particularly the part of your body which we are born with. And they include physical barrier and the blood-borne mechanisms and the cells, systems. So in physical barrier, for example, your skin, mucous membrane, saliva, flushing action of urine and tears, stomach acid. These physical barriers act against all kind of microorganism. They are not specific against one particular mechanism or microorganism. For example, your skin, tough skin is boundary against virus, bacteria, physical trauma, chemical trauma, radiological trauma, all kind of trauma to the body, including the microorganism, okay? So for example, skin is tight, so it prevents microorganism to get inside. Mucous membrane contains a lot of enzymes and mucus which clears the microorganism. For example, when you inhale virus or bacteria in nose has mucus and that mucus clears microorganism. Saliva in your mouth contains enzyme that breaks down microorganism. When you pee, the flushing action of your pee clear any microorganism attached in your urethra. When you swallow or eat food with microorganism in your stomach, pH is very low, which is pH 1, highly acidic, and that kills the microorganism immediately. So this physical barrier stops infection before it enters the body. But what happens? Sometimes microorganism can breach the boundary of physical barriers and can enter into the body. And then once it enters the body, it is the immune system of your blood which start fighting. But this immune system inside your blood, inside your body, is also called innate immunity because it is not specific against particular microorganism. These blood-borne mechanism fight against all kind of microorganism like bacteria, viruses, protozoa, fungus, and other parasites. That's why even they are fighting and working inside your body in the blood, we still call it innate. And that innate systems are blood-borne, and there are two types. One is phagocytes, they are the cells because sites, and another is complement cascade system. So phagocytes are white blood cells which by chance after the breach of physical barrier enters your body. These are the cells which fight infection because they start killing and eating those microorganisms. And these phagocytes means microorganism eaters are neutrophils, macrophages, basophils, eosinophils, and natural killer cells. These white blood cells cause death of dangerous organism which has entered your body. Okay. The function of these phagocytes is also activated by acquired immune system. We will talk about that later. Okay. The next part is complement cascade. Complement are a kind of protein molecule found in our plasma, in our blood. And there are several kinds of complement. So when there is any microorganism invasion in our body, these complement system get active and they change their structure. And then they clear the microorganism, induce our immunity, make microorganism weaker, and produce non-specific immunity. There are several pathways. There is alternative pathway, direct pathway, indirect pathway of the complement system, and we're not going to talk about that much. So this alternative pathway then ultimately through the natural killer cells or through the white blood cells, particularly T lymphocyte, they directly kill the microorganism or they activate the phagocytes. Okay, so this is immune system. Now, when these phagocytes get exposed to microorganism, not only they kill that microorganism, they also share what kind of microorganism was with the acquired immune system. So let's see how that happens. When these macrophages and other antigen presenting cells, antigen presenting cells, expose to microorganism, they take their protein molecules, which is called antigen, and then they present to the lymphocytes. Okay, and when lymphocytes deal with them and produce immunity, particularly to that individual antigen, then we call it acquired immunity. So acquired immunity is after the exposure to the microorganism. Okay, so acquired immunity, once these white blood cell or any other antigen presenting cells in the body present antigen from the microorganism to the T cell or B cell, then we call it acquired immunity. That's why acquired immunity is the function of T cell and B cell. And acquired immunity is divided into two groups. That is T cell immunity, which we also call cell mediated immunity, or B cell immunity, or we call it humoral immunity. When T cell or T lymphocyte exposed to antigen, whole T cell release into the three kinds of cells. That is called suppressor T cell, helper T cell, and cytotoxic T cells. And then these cells cause death of the body's cells that are infected with virus or otherwise damaged cells. That means active T cell attack the infected cell directly, attack the cancer cell directly, attack any abnormal cell in the body directly. Whereas B lymphocyte produce humoral immunity. That means once B cell exposed to antigen, they produce their clone and divide and produce a lot of baby white blood cell called lymphoblasts. And then these lymphoblasts change into two kinds of B cells, plasma B cells and memory B cells. Memory B cells keeps the memory of that particular antigen, whereas plasma cells produce antibodies. These antibodies then activate the complement protein, and that then activate classical pathway, which pathway activate ultimately the innate immune system. So this is the bridge between the innate and acquired immunity. That means there is the correlation between the innate and immune system. Innate system does some function, and if innate system fails, then it gives to the acquired immune system to work immediately and to keep the memory for the future. This is the summary of innate immune system. Okay, so let's go now and talk about in detail the innate or non-specific immunity. Non-specific immunity or any immunity is our body's defense mechanism. So defense mechanism is the immune system, or the immune system is the structure and processes against the pathogen. So any structure or processes in our body, for example, our saliva and processes like example phagocytosis are the defense mechanism or immune system can be grouped into two categories. As you know, they are innate or non-specific immunity, or adaptive or specific immunity. Innate or non-specific immunity is inherited as part of structure of each organism and comprises all the white blood cells except lymphocyte. So innate immunity is the part of your body as well as all white blood cell except the lymphocytes. Whereas adaptive or specific immunity is particularly the function of lymphocyte. And these adaptive or specific immunity change only with exposure to particular microorganism. That means their antigens. So again, if you compare innate and adaptive immunity, you see here in the left side you have innate immunity and your right side here adaptive immunity. So here you have epithelial barrier, one of the innate immunity example. Then you have phagocytes, you see dendritic cells, the phagocytes, large eating cells. When there is damage, these dendritic cells or phagocytes get the antibodies, process them, then they activate complement system, activate natural killer cells, and they attack the microorganism or cells infected with microorganism. And then these phagocytes and dendritic cells also present antigen to B and T cells, okay? So that becomes adaptive immunity. So B cell, when exposed to antigen, you see here, they turn into plasma cell which produce antibodies, and T cell cannot recognize antigen directly. So these antigen presenting cell, you see here, this antigen presenting cell is taking the antigen from microorganism and presenting to the T cells. And then T cells becomes cytotoxic, helper, regulatory T cells. And that effector T cells then attack the infected cell or abnormal cell or cancer cell and produce the immunity, okay? So this complement system has four major functions. That is the lysis of infectious microorganism. When this complement system is activated, it lyse the microorganism, means kill the microorganism. They also activate inflammation process, increase the inflammation. That means they activate the histamine producing cells, like the mast cell. They produce opsonization, means they activate antibodies to attach to the antigens and clear the immune. So those are the function of complement system, okay? If you see here time-wise, time after infection, innate immunity works at the beginning. So like, sorry, innate immunity is like your first line of defense. And the adaptive immunity is like the inner core, hard defense mechanism, okay? So like local sheriff is innate, and your FBI or Central Reserve Army, those are our adaptive immunity, okay? So let's talk about adaptive immunity. So how do cells of innate immune system recognize invaders? So if any microorganism, when it enters our body, how they recognize our immune cells, whether it is self or non-self. That means whether it is insider or outsider. Like our body produce protein and other component in millions of trillions of number every day, every moment. But those proteins are not considered as foreigners, and our immune cells do not attack them. But when they are foreign, they attack them. So how they recognize them? They distinguish between self and non-self by recognizing molecules called PAMPs, PAMPs, which is pathogen associated molecular pattern. Okay. Immune cells has these receptors, and that receptors can recognize whether it is self or non-self. Innate immunity is the first line of defense against invading pathogens. Pathogens means disease-causing agents. And those innate immunity defense are epithelial barriers, high acidity of your gastric juice, engulfing the microorganism, which is called phagocytosis, interferons produced by virus-infected cells, fever when there is high temperature of body in response to pyrogen produced by your white blood cells, and other function of white blood cells except the lymphocytes. So these all this list are the parts of innate immune system. Okay, so our immune cells plasma membrane contains the receptors, and those receptors can recognize whether a protein inside our body is from our own cells or it is from outside, like microorganism. So usually the microorganism has a PAMPs, which is called PAMPs, pathogen associated molecular patterns. There are two kinds of PAMPs on the microorganism, common PAMPs. They are lipopolysaccharide, LPS, from gram-negative bacteria, and peptidoglycan from gram-positive bacteria. Apart from lipopolysaccharide and peptidoglycans, there are some other PAMPs found on the microorganism, and they are flagella, foreign DNA, and RNA. Okay. For example, coronavirus DNA is antigen, and that is called PAMPs, and that antigen is recognized by our immune cells. Okay. Some of our immune cells have receptors for PAMPs displayed on their surfaces. So the receptors are called toll receptors. So toll receptors on our immune cell plasma membrane recognizes PAMPs like LPS, peptidoglycan, flagella, foreign DNA, and RNA. To recognize these PAMPs, we have 10 different kind of toll receptors. Okay. So once the physical barrier of our body is breached, damaged, and the microorganism get inside, then our immune cells, white blood cells, start engulfing and phagocytosing them. And that process is called phagocytosis. So phagocytosis is eating the microorganism. And phagocytosis is performed by 3 classes of phagocytic cells, or 3 kind of white blood cells. They are neutrophils, mononuclear phagocytes, which is called macrophage and monocytes, and then organ-specific phagocytes. That means the big eaters found in liver, spleen, lymph nodes, lungs, and brain. Sometimes these organ-specific phagocytes live permanently inside the organ, and then we call them fixed phagocytes. For example, in the liver, we have in the liver sinusoids, we call them Kupffer cells, in the spleen, in the lymph nodes, and they remove pathogens. So these phagocytes start engulfing the microorganism when they enter our body. So what is the process for phagocytosis? As you see here, when you have a cut or damage, the bacteria enters your skin or your mucous membrane. Once it enters, then white blood cells come and they produce their the pseudopods and take bacteria inside. Okay, so in the connective tissue, blood contain white blood cells. This is white blood cells, WBC. These white blood cells are attracted by chemotactic factor, and that process is called chemotaxis. And then bacteria, the neutrophil exit the blood vessel, which process is called extravasation or diapedesis, and they come out and swallow the bacteria. Okay, let's see what happens once they swallow the bacteria. So one white blood cell or neutrophil swallow this bacteria. You see, bacteria get inside and then they mix with lysosomes, okay, and form the vacuole. So take the bacteria, take the inside the lysosome and keep inside the lysosome and start cooking it, digesting it. So pseudopod forms phagocyte surround pathogen, forming a vacuole. Vacuole feeds with lysosome, which digest pathogen and kill the microorganism. Okay. At the same time, these white blood cell and other phagocytes produce pyrogens. They produce pyrogens. Pyrogens. They produce pyrogens. And that pyrogens then produce fever. Fever is produced in response to pyrogen to increase the body temperature in hope that in high body temperature, microorganism will not grow and multiply. Okay, that's why fever, which is increased body temperature, is the component of innate immunity, because fever can suppress any kind of microorganism. How we produce fever? It occurs when hypothalamic thermostat is reset upward by interleukin 1b and cytokines. These interleukin 1 beta and the cytokines, which is collectively called endogenous pyrogens, means pyrogens from your body. These two stuff, pyrogens, keeps your hypothalamus up. Hypothalamus is like our thermostat, like we have air conditioned thermostat in our house. Our body thermostat is hypothalamus, and the pyrogens set it upward. So it increases your body temperature in hope that it will kill microorganism. Okay. So pyrogens are released by white blood cell in response to endotoxin from gram-negative bacteria, and they induce fever by producing endogenous pyrogens. And at the same time, these pyrogens or cytokines also increase sleepiness. That's why when you have inflammation, infection, you feel sleepy. Okay. Another component of innate immunity is interferons. So interferon are polypeptides produced by the cells infected with the virus that provides short-acting, non-specific resistance to viral infection in nearby cells. And they are three types: alpha, beta, and gamma interferon. So when you have infection with a virus, the virus enters your cell and control your nucleus and control the function of that cell and produce more and more virus. And that virus then release from that particular cells and infect other cells. But at the same time, our nearby normal cells in our body start producing a protein, and that protein is called interferons. And those proteins then covers the normal nearby healthy cells and protect from that nearing bacteria, viruses. And that is interferon. So interferon protect healthy cells from virus infection. And this is pretty much kind of all kind of virus infection. So let's see, if you get infected with coronavirus now, your body will be producing interferons, and that interferons will protect you from the coronavirus, try to protect. If coronavirus is stronger, it will infect more and more cells. And at the same time, interferons will also protect you from other virus infection. That's why it is the part of non-specific immunity. Okay. So here, effect of interferons. When interferon is produced, it has two kinds of function: stimulation and inhibition. It stimulates macrophage phagocytosis. You know, in non-specific immunity, macrophage and phagocytosis was a process. So it stimulates macrophage phagocytosis. It increases activity of T cells, particularly cytotoxic killer T cells. It also activates natural killer cell function, and it helps in production of antibodies by plasma cells. At the same time, interferon has some inhibition function, like it inhibits the cell division, it inhibits growth of tumor, it also inhibits maturation of adipose cells and maturation of red blood cells, erythrocytes. Okay, so we are done with the non-specific or innate immunity. So innate immunity is, again, a physical barrier and the immune cells inside your tissue or in your blood. Once immune cells in the body or in your blood start acting, that's the time adaptive immunity starts. But that is still the part of non-specific immunity. When B and T cell starts its job, then adaptive immunity starts, okay? So adaptive immunity is acquired ability to defend against specific pathogens by prior exposure to those specific pathogens. And it is mediated by production of specific antibodies by lymphocytes. So adaptive immunity is possible only when you expose to that particular antigen and when your T cells and B cells are active, okay? So that means to produce adaptive immunity, your lymphocyte must expose to antigens. And antigens are the part of microorganism or part of any foreign particles or part of abnormal particles inside our own body. That is antigens. So here, let's define the antigens. Antigens are molecules that elicit production of antibodies that specifically bind those antigens. Antigens are usually large molecules that are foreign to the body. Our immune system can distinguish self molecules from non-self antigens, and normally they becomes active and makes antibodies only against non-self antigens. Large complex molecules can have a number of antigenic determinant sites, different sites that stimulates production of and bind to different antibodies. So basically antigens are protein molecules, and our immune cells can distinguish whether it is foreign or self. Some of the antigens are large and they have several antigenic determinant sites. Some of the small molecules in our body are non-antigenic molecules. We call them haptens, okay? But they become antigens when bound to other protein, particularly from an antigenic determinant site. So let's see if there is a large molecule and there is several antigen, and if this hapten binds with that antigen, then it becomes antigenic. Otherwise, these hapten molecules are non-antigenic. These haptens are used for creating antibodies for research and diagnosis usually. Okay. So when antigen and antibody binds with each other, they agglutinate, and that process is called agglutination. And when we have to detect any antibodies in your body, we do an assay, and that assay is called immunoassays. Okay, so how we do, look here in the diagram. Here we have these Y-shaped immunoglobulins or antibodies, and we have latex particles. These antigens are okay. So antigen X is here, and these are the anti-X. The Y-shape is anti-X antibodies. So when this X antigen binds with this Y antibodies, then what happens? These all latex with antigen and antibody complex start making clumps, and that clumping of antigen and antibodies is called agglutination. And when we tag this antigen and antibodies with some fluorescence light and put under microscope, you can see the blue color light coming out of it. And that is called the immunoassay. So let's see if you are exposed or not. Like some people exposed to coronavirus but do not produce any symptoms. That doesn't mean you don't have immunity. Your body will produce immunity against coronavirus and you become immune to coronavirus. Can we check that? Yes, by this method. We're going to take outside made antigen, and then we will take antibodies from we will take our blood, take our white blood cells, take our plasma, the clotting factor, and take some serum from our blood, and then mix with the antigens, which is made in the lab from the corona DNA, and mix it. If they make clumping, that means you have antibodies. If they don't, that means you don't. And that's the way the antibodies tests are done. Okay, so this is all about the first part of the immune system. Again, immune system can be classified into two groups: the innate immune system and the adaptive immune system. innate and adaptive immune system work together to protect us from the foreign antigens, particularly foreign microorganisms or the foreign abnormal antigen produced in our own body, like in the form of cancer cells. Okay, so this is the first part of immune system, and we will talk about the adaptive immunity in detail in next lecture. Okay, thank you.